It's time for another warm-up. Let's get going. Which muscles and nerves are derived from the second branchial arch? Well, the mnemonic for the second branchial arch is the letter S. So you have cranial nerve 7, so that's the facial nerve, and that innervates the muscles of the facial expression. And then the S muscles include the stapedius and the stylohyoid. Next, what is the antidote for each of the following substances? So first we have opioids, so either naloxone or naltrexone. Antidote for benzodiazepines is going to be flumazenil. Tricyclic antidepressants, you're going to alkalize the urine with sodium bicarbonate to help get rid of those. For heparin, the answer is protamine. Warfarin interferes with your liver's ability to make the vitamin K dependent coagulation factor, so the reversal agent is vitamin K. An immediate, though, reversal of warfarin effects, you want to give things like fresh frozen plasma to replace those coagulation factors if the patient is deficient. And for theophylline overdose, the antidote is beta blockers. Next, which ECG finding is associated with hyperkalemia? So remember, T waves mirror the potassium level. So with high potassium, you get peaked T waves. With low potassium, you get flattened T waves, maybe even a U wave as well. All right, guys, that's it for the warm-up. Let's get to that lecture.
Before we launch into antigen presentation, I want to take just a minute to address my reputation around here. Everybody thinks of Dr. Jenkins as the founding father of DIT, and Dr. Lewis as the funny one, and Dr. Schufert as the smart one. But what you may not know is that I'm sort of the bad boy of DIT, the loose cannon, always laughing in the face of danger. So, you know those little packets of desiccant that say, "do not eat"? I eat them. You know those tags on pillows that say, "do not remove under penalty of law"? And what about this warning on matchbooks? Close cover before striking. We'll see about that. You're not the boss of me. Ow! Maybe there's a good reason for that one. All right, let's talk about antigen presentation. What exactly does antigen presentation mean? To recap what we said earlier, a phagocyte, like a dendritic cell, engulfs some pathogen, like a bacteria or a virus or whatever, and it digests it, and then the dendritic cell puts a little piece of that pathogen on the outside of the dendritic cell, where helper T cells can see it and bind to it. So dendritic cells are antigen-presenting cells that are presenting a foreign antigen to the helper T cells. And the whole point is to activate the helper T cells, to tell the immune system that there's some invader that they need to start attacking. So we're going to talk about the different kinds of T cells and how helper T cells get activated in another video. But for right now, I just want to go over a couple of really basic points about T cells that apply to antigen presentation. T cell precursors begin life in the bone marrow, and then they travel to the thymus where selection takes place. And the cells that are selected mature into T cells, and the rest that aren't selected are destroyed. Now, we're going to talk about all that later. But remember, they're called T cells because they mature in the thymus. T for thymus. Now, a T cell is a type of lymphocyte, but you can't tell a T lymphocyte from a B lymphocyte just by looking at them. So, how do you tell them apart? Well, you can tell them apart by the different surface markers and receptors found on the T cell. And these surface markers are worth knowing for step one. So every T cell has a specific T cell receptor, or TCR, that's capable of binding to a very specific antigen. Remember, T cells are part of the adaptive immune system, which is very specific. So T cells have T cell receptors that can bind to a specific antigen. And actually there are hundreds or maybe thousands of T cell receptors on the surface of a single T cell. All those receptors are identical. Now, this T cell might have thousands of receptors for an antigen that's found on the chickenpox virus or the varicella virus. This other T cell has thousands of receptors for a measles virus antigen, and so on. Now, very closely associated with this T cell receptor is a surface marker called CD3. Now, CD3 actually helps the T cell receptor transmit a signal into the T cell when it binds to its specific antigen. So I want you to think about CD3 as a marker for all T cells. Now, just in case you thought this was getting too simple, there are different types of T cells, which can also be differentiated by their surface markers. We mentioned helper T cells. Helper T cells have a CD4 surface marker. They're sometimes abbreviated TH, so sometimes they get called T helper cells. Helper T cells or T helper cells, same thing. Then the other big type of T cells are the cytotoxic T cells, which are abbreviated TC. Now, cytotoxic T cells are identified by a CD8 surface marker. There are also memory T cells and regulatory T cells, which used to be called suppressor T cells, and a few other types of T cells that you don't really need to worry about for step 1. Again, the term lymphocyte refers both to B cells and T cells, but around 80% of the lymphocytes in circulation are T cells. So when somebody talks about lymphocytic infiltrate, for the most part we're talking about T cells. Let's talk about how helper T cells get flagged down by an antigen-presenting cell. Basically, the T cell receptor on a helper T cell will bind to its specific antigen only when that antigen is presented by an antigen-presenting cell. So this is because the CD4 marker on a helper T cell is actually very closely associated with that T cell receptor. And the CD4 marker actually links up with a protein complex on the surface of the antigen-presenting cell, which is called a major histocompatibility complex, or MHC, specifically MHC class II. Now the way this works is after the antigen-presenting cell phagocytoses that pathogen and digests it, puts a piece of the antigen on its surface attached to an MHC2 molecule. Then that antigen-presenting cell can start presenting this antigen to every T cell it meets, looking for the one with matching T cell receptors. Now, when it finds one, the CD4 on the T cell has to bind to the MHC2 on the antigen-presenting cell. And when that happens, the CD4 and CD3 can start their signaling cascade that ends up activating the helper T cell. We're going to talk about what activated helper T cells do in another video. But for now, just remember that CD4 positive helper T cells bind to MHC class II. Now in the video, when we talk about T cells, we're also going to talk about cytotoxic T cells. Now, cyto means cell, so what do you think a cytotoxic T cell does? It kills cells. Specifically, cytotoxic T cells are supposed to kill virus-infected cells or cancer cells. Well, that's all well and good, but how can the cytotoxic T cells recognize virus-infected cells or cancer cells? How can they tell the difference between a healthy cell and a diseased cell? Well, the answer is the major histocompatibility complex again, but this time it's MHC class I. So MHC class I is expressed by basically every nucleated cell in your entire body. So not the red blood cells, but pretty much every other cell in your body expresses MHC class I molecules on their surfaces. And attached to those MHC1 proteins are peptide antigens that come from inside the cell. So your antigens are slightly different from my antigens. So those unique antigens get attached to something called the peptide binding groove on the MHC class I molecules on the surface of all your cells. And it's kind of a way for your cells to tell the cytotoxic T cells, hey, leave me alone, I'm on your team, I'm one of the good guys. But when a cell is infected by a virus, it starts putting viral peptide antigens in that peptide binding groove of the MHC1 molecules. And that tells the cytotoxic T cells, psst, virus, I got virus in here. And then the cytotoxic T cells are going to recognize that viral antigen and kill the cell. Because remember, a virus-infected cell is just a factory for more viruses, and the only way to deal with it is to kill the infected cell. It's the same kind of thing with a cancer cell. Cancer cells start expressing weird cancer antigens on their MHC1 molecules, and that tells the cytotoxic T cells that something's up, so that those cytotoxic T cells can come attack the cancer cell and kill it. So think back. If CD4 was the surface marker for helper T cells, what do we say the marker was for cytotoxic T cells? CD8. So just like CD4 binds to MHC class II on antigen-presenting cells, CD8 binds to MHC class I on virus-infected cells or on cancer cells. And then the cytotoxic T cell can kill those diseased cells. So it's pretty easy to keep all this straight in your head. You just multiply the CD number by the MHC number, and the answer is always 8. CD4 times MHC2 equals 8, and CD8 times MHC1 also equals 8. How convenient is that? Now let's dig into some genetics of the major histocompatibility complexes. Like all proteins, MHCs are derived from very specific genes. The genes that code for the MHCs are called the HLA genes, human leukocyte antigen genes. And just like your MHC1 molecules are unique to you and different from my MHC1 molecules, your HLA genes are different from mine. Again, MHC1 molecules are found on virtually every nucleated cell in your body, and they're coded for by 3 genes, HLA-A, HLA-B, and HLA-C. Then HLA-D codes for MHC2, but we break those down into 3 subgroups. Remember, MHC2 molecules are only found on antigen-presenting cells, and the genes that code for these MHC2 proteins are HLA-DR, HLA-DP, and HLA-DQ. Now, all these HLA subtypes can be further classified into specific loci and specific alleles. So you might divide HLA-DR into HLA-DR1 or DR2 or DR3 and so on. Or for HLA-B, you can have HLA-B1 or HLA-B2 and so on. That's where HLA-B27 comes from, and that should sound somewhat familiar to you. Now, the importance of this is that there are certain alleles and certain loci that are associated with certain diseases. And not too surprisingly, these are generally autoimmune diseases, where the patient's own immune system is attacking from within. Now, there are just a couple of these diseases that are most likely to show up on step 1. This is about a 2 or 3 star topic. Not all of you are going to have a question on the association between HLA and certain diseases, but a few of you will. The important ones you need to know about are HLA-B27 and HLA-DR3 and DR4. Now, as we discussed in rheumatology, HLA-B27 is associated with the seronegative spondyloarthropathies. And the mnemonic for these diseases is PAIR. So the P is for psoriatic arthritis, the A is for ankylosing spondylitis, the I is for the arthritis you can sometimes see with inflammatory bowel disease, and then the R is reactive arthritis, or the older name was Reiter syndrome. And then HLA-DR3 and HLA-DR4 are associated with type 1 diabetes, which is caused by autoimmune destruction of the pancreatic beta cells. Cells that express MHC2 on their surface are antigen-presenting cells. Now, there are a handful of cells that can act as antigen-presenting cells, like macrophages and sometimes certain B cells, but by far the most important type of antigen-presenting cell are the dendritic cells. Now, let's look at number 4 in your study guide. They're called dendritic cells because they're covered with these membranous extensions that look like the dendrites on a neuron. They've got their feelers out and they're trying to capture any antigen that might come along. Dendritic cells display class 1 and class 2 MHC molecules. They display class 1 because all nucleated cells display MHC class 1, and they display class 2 because that's how they present antigen to the helper T cells. They also have a surface protein called B7, which can then bind to specific proteins on the T cell receptor, and that will either increase or decrease the signal that's activated when the T cell receptor binds to the antigen that's being presented. So B7 is sometimes called a co-stimulatory molecule. It won't activate the T cell by itself, but it can influence the activation of the T cell when other signals are around. Dendritic cells also have a CD40 surface marker, which allows the T cells to activate the antigen-presenting cells. So everybody's stimulating everybody else. Now, we're going to see CD40 and B7 again in the next video. This is just a brief introduction. But CD40 is found on dendritic cells. It's also found on other antigen-presenting cells, like macrophages and B cells. So, what do dendritic cells do? Well, they're designed to capture antigen. So they capture antigen at one location, and then they present it to T cells at another location. Once a dendritic cell captures an antigen, it changes shape. It's no longer a cell with those long dendritic arms. It changes shape so that it can migrate to the lymph nodes and say, hey, look what I found. I found some antigen. Now, out in the periphery, dendritic cells serve as peripheral sentinels. A sentinel is like a guard or a watchman or a sentry. Now, this is sometimes called the immature state or the sentinel state. And in that sentinel state, that antigen capturing state, they can capture antigens by 3 different mechanisms. By phagocytosis, by receptor-mediated endocytosis, where the dendritic cell's surface receptors can recognize an antigen and then endocytose that antigen using clathrin-coated pits. And then the third mechanism is called pinocytosis, which is like endocytosis of extracellular fluid and whatever small particles are floating in the fluid. Kind of like cellular drinking of fluid. Dendritic cells can actually replace their entire cell volume of fluid within an hour through pinocytosis. So it's kind of like a sieve. It's just taking in a bunch of stuff and looking for any antigen that it can find in that fluid. So the cell starts in this immature sentinel state, this antigen capturing state. And then once it's captured an antigen, it makes a transition to an antigen presenting state, which allows it to travel into the circulation. And in this state, the dendritic cell has maximal capacity to present antigen to helper T cells because it has increased expression of class 2 MHC and increased expression of those co-stimulatory molecules. So they find an antigen, then they switch to this cell that presents very well to helper T cells. There are also different types of dendritic cells. One type you may have heard of is the Langerhans cell. A Langerhans cell is a dendritic cell that's found in the epidermal layers of the skin. Sometimes this is referred to as cutaneous associated lymphoid tissue, or CALT, C-A-L-T. So the Langerhans cell is a type of dendritic cell that hangs out in the epidermis. And again, once it finds antigen, it's going to change shape and migrate to the lymph nodes. And then in the interstitial spaces of the organs, except for the brain, you have interstitial dendritic cells. Let's briefly discuss a pathology that can arise in the Langerhans cells, which is called Langerhans cell histiocytosis. This is a disease where there's excessive proliferation of Langerhans cells, these dendritic cells in the epidermis. And then the problem is that these cells are no good at antigen presentation, so they're not really able to do their job. And these cells express an S100 tumor marker and also a surface marker called CD1A. And the way you'd probably see this on your test is that on electron microscopy, these cells are going to have these Burbeck granules, which look like tennis rackets within the cell. All right, that's it for antigen presentation. So pause the video and try to answer those end of session quiz questions. All right, first question. What molecules are expressed on the surface of dendritic cells? Well, we mentioned MHC class 1, MHC class 2, a B7 protein, and CD40. Next, what are 3 cell types that are known for presenting antigens to CD4 positive T cells? We talked about the dendritic cells, then also macrophages and B cells. Next, what characteristic finding would you see on electron microscopy of a dendritic cell in a patient with Langerhans cell histiocytosis? That was the Burbeck granules. And the last question, which MHC class is recognized by helper T cells and by cytotoxic T cells? So helper T cells recognize MHC class 2, cytotoxic T cells recognize MHC class 1. All right, that's it for the end of session quiz. I'll see you next time.